The overall goal of this procedure is to study the molecular mechanisms underlying learning and memory formation in honeybees. The first step is to collect free-flying bees from their hive. Next, the bees are harnessed to plastic tubes. The third step of the procedure is to perform classical conditioning. Drugs can be injected at certain time points around conditioning in order to study their effect on learning and memory formation. The final step of the procedure is to test memory retention. Ultimately, changes in behavior after drug treatment may reveal the molecular mechanisms underlying learning and memory formation in honeybees.
This method can help answering key questions in the field of learning and memory by uncovering the molecular mechanisms of memory formation.
In general, researchers new to this method will have difficulties because the timing of the stimuli presentation during conditioning and memory retrieval must be exact. Also, the feeding regime is critical.
The procedure will be demonstrated by Katrin Gehring and Johannes Felsenberg, two graduate students from my laboratory.
On the day before the experiment, collect bees between 2 and 4 p.m. using a UV light permeable plexiglass pyramid. Once back in the lab, place the pyramid on its base and darken the walls with a towel, leaving the apex uncovered. Open the apex and hold a glass vial over the opening, allowing a single bee to travel into the vial before reclosing. Continue this